Hi, my name is Noam. I'm excited to take part in this symposium about the wonderful cerebellum. In this talk, I will share with you our findings about the organization of the human cerebellum, which we estimated within the individual. The cerebellum is long known for its somatomotor function. In fact, until not so long ago, it was considered a structure that is involved only in somatomotor processing. So, what do we know about the somatomotor cerebellum? 80 years ago, with electrophysiological recordings, Adrian discovered that body parts are topographically organized in the anterior lobe in an inverted fashion. A year after, Snyder and Stoll confirmed the anterior lobe somatomotor topography and discovered a second body map in the posterior lobe that was upright rather than inverted. The anatomical basis of the two separate somatomotor maps was revealed half a century later, and the two maps were repeatedly observed in humans using non-invasive neuroimaging techniques. Beyond these two established body maps, there are hints of a third somatomotor representation in the literature. For example, Kaufman et al. Showed that the cerebellar vermis is a target of projections from motor regions in the cerebral cortex. So, is there a third somatomotor body map? A definitive demonstration of one, if exists, faces multiple challenges. First of all, it is in a potential small area within the already small cerebellum. It is expected to have lower SNR, and it's potentially adjacent to the posterior map. So to face these challenges, we would want a great amount of power. We would want to take the within-individual approach to minimize spatial blurring, and also to have multiple body parts representation to be able to separate potential adjacent maps. So, in our experimental design, participants performed a block design task paradigm consisting of 6 types of movements: right and left hands, right and left foot, tongue, and glutes. What we basically did is we overwhelmed with signal. Each participant had 4 sessions with 6 runs in each, adding up to 576 separate blocks of body movements per participant. As a sanity check, on the right is a coronal slice of the cerebral motor cortex where body topography is evident, with the tongue in red most laterally, the hand in yellow, and then the glutes in green, and the foot in blue most medially. The data also clearly show the full topography of the known anterior and posterior body maps in the cerebellum. While it seems there is something in the vermis next to the secondary map, the variability across participants and the adjacency to the posterior representation makes it still uncertain. So, to further explore, we focused specifically on hand and foot movements where direct contrast between the right and left movements were possible. For the hand, the anterior and posterior lobe representations are clear and distinct. It seems that there is another representation on the edges of the vermis, although it is unclear if it's a distinct third representation or simply a continuation of the second representation. Contrasting right and left foot movements yielded robust responses that also included a representation in the vermis which is spatially distinct from the 2 others. Functional connectivity from seed regions in the third somatomotor representation confirmed anatomically specific connectivity with the cerebral motor cortex, paralleling the patterns observed for the 2 well-established maps. Importantly, the region between the 2 foot representation is associated selectively with the hand, providing evidence that the 2 foot representation are parts of spatially and functionally distinct maps. A third foot representation was found in each one of our 7 out of 8 highly sampled individuals. So now, after localizing the somatomotor representation in the cerebellum, the big question is, what lays between these somatomotor maps? So in the past 30 years, studies are accumulating to show the involvement of the cerebellum in cognition. To uncover the detailed functional organization of higher order processing, we again overwhelmed with signal within the individual. We have 15 participants, 8 to 11 MRI sessions per participant, including 120 to 205 minutes of resting state data, and we also acquired data from multiple tasks based on many previous studies to specifically target hypothesized processing. We estimated 15 networks in the cerebral cortex using a multi-session hierarchical Bayesian model, and then, based on functional connectivity, we derived the cerebellar parcellation, here shown for one representative participant. As expected, the region of the somatomotor maps are occupied by the two somatomotor networks of the cerebral cortex. In between, the region of the Crus I, Crus II lobules is occupied primarily by 5 higher order networks. We will now focus on this region and these 5 networks. A useful visualization to better orient is the inflated cerebellar cortex, shown here in a posterior view. For 3 subjects, we see the inflated cerebellar cortex with the 5 networks overlaid. The default networks, DNA and DNB, the frontoparietal networks, FPNA and FPNB, and the language network, LANG. These 5 networks cover Crus I and Crus II lobules and extend to the adjacent lobules. Each network possesses multiple regions that are closely juxtaposed with other networks, creating an interdigitated topography. The exact spatial positioning and extent of network representations varies across individuals, but the general organization is conserved. For example, the most posterior region of Crus I and Crus II lobules is occupied by DNB. It is bounded by FPNA and FPNB medially and laterally, and by the language network both anteriorly and posteriorly. Here are all 15 participants, again showing the same general organization with some idiosyncrasies. Given this detailed topography, we asked how specific these network assignments are. So we used seed-based functional connectivity as an independent method, free from prior assumptions. Here, a seed in the cerebellar DNA network and the corresponding DNA in the cerebral cortex on the right. Looking at the functional connectivity pattern in relation to the borders of the network showed that each seed recapitulated the full distributed extent of the relevant network in the cerebral cortex, demonstrating a remarkable specificity of the 5 networks in the Crus I, Crus II extended zone. Here is also for DNB, for FPNA, FPNB, and for the language network. We also saw high specificity is evident for the multiple representation of the same network. For example, here another language region. So with this remarkable specificity, what are the functional characteristics of these networks? Are they dissociable from one another? We constructed hypotheses driven by functional properties of the 5 networks in the cerebral cortex. First, we defined regions of interest, as each network within the Crus I, Crus II extended zone after excluding voxels with very low SNR and low confidence of the network assignments. We first targeted the frontoparietal networks FPNA and FPNB with a load effect paradigm, the well-known NBAC task, contrasting a 2-back high load condition with a 0-back low load condition. For example, here on the right, depending on the block type, a different face would be a match for the 0-back and for the 2-back. The bar plot on the right shows the average Z values within an ROI, average across all 15 participants. FPNA and FPNB ROIs were merged together as our a priori hypothesis was that both networks will similarly respond to load manipulation. Indeed, the response to the 2-back versus 0-back contrast in the merged ROI of the frontoparietal networks was found to be significantly greater than 0. Although DNA and Lang ROIs were also significantly greater than 0, the response in the frontoparietal ROI was found significantly larger than the response in all other ROIs. To test whether the response was driven from a specific type of stimulus, we quantified the average response to multiple stimulus type. The experimental paradigm, along with faces, included 0-back and 2-back blocks of letters, of words, and pictures of scenes. The average z-value for all stimulus types in the FP&A FP&B merged ROI was found significantly greater than 0. These results suggest that the FP&A and FP&B region exhibit a domain-flexible response to task load. So, what about the other networks within the Crus I, Crus II extended zone? Are these 3 networks regions domain-specific? With what we know about their counterparts in the cerebral cortex, we targeted these networks with specific tasks and quantified the average z-values in each ROI across participants. First is the episodic projection task, which targeted DNA network, driving the participant to remember their past and imagine their future. Next, we explored the response to theory of mind tasks, which targeted the DNB network. We adapted two tasks paradigm to relate the participants to other people's mental state. Lastly, to target the language network, we utilized a sentence processing task. Quantification of the response with the ROIs revealed a significant 3x3 interaction, where DNA ROI significantly responded to the episodic projection task contrast, while DNB and Lang did not. DNB ROI significantly responded to the theory of mind task contrast, while DNA and Lang didn't. The sentence processing task contrast evoked a significantly greater response in the Lang region than in DNA region, and greater but not significant than in the DNB region. Taken together, these results suggest a triple dissociation between DNA, DNB, and Lang region within the Crus I, Crus II extended zone, as each region shows preferential recruitment to a specific cognitive task contrast. With that, I would like to conclude. I hope I provided evidence to convince you about the existence of a third somatomotor representation in the vermis. There are still remaining gaps. For example, what is the orientation of this third map? We further dived into the Crus I, Crus II extended zone, which is occupied by 5 higher order networks that are closely interdigitated with one another. Using task data, we introduced 2 types of functional dissociation. One is between the frontoparietal domain flexible networks to the other 3-domain specialized network. The second is a triple functional dissociation between DNA, DNB, and language regions across episodic, social, and language domains. I would like to thank my great mentor, Randy Buckner, the entire Buckner Lab, specifically Lauren Di Nicola, Peter Angeli, Heather Kosakowski, and Jingnan Du that contributed a lot to these projects. The team of the Harvard Center for Brain Sciences, to our funders, and to all of you for watching.